Okay, so another reason humans don't make good experimental organisms, raising humans is expensive. They need food, love, stimulation, maybe even some college tuition. Research funding is limited, so model organisms are ideal because they are cheap to grow. For example, fruit flies can be grown by the hundreds for a few dollars worth of fly food in a tube. Mice are more expensive because they eat caviar and champagne. Now, okay, they don't eat caviar and champagne, but they are kind of costly. However, sometimes they're the best way to study animal-specific processes, behaviors, and especially diseases. Model organisms must also grow well under controlled lab conditions. So a cave beetle that requires dark and humid conditions would probably not make the best model organism. Working with inexpensive and adaptable organisms allows us to run more experiments more quickly with less money. Oftentimes, when we want to do an experiment in genetics, we take a group of individuals, split them into two groups, and change something about one of the groups, like giving them a drug. And then we don't do anything to the other group, which serve as controls. However, individual humans are very different from each other, even genetically. Ideally, in an experiment, all the individuals are the same, at least at the beginning of the experiment, so that if any changes are observed, you can be confident that those changes are caused by the thing that you changed about one of the groups, in this example, the drug. If individuals are genetically and phenotypically diverse, if you do the same experiment, results become difficult to interpret.
This is because any difference seen between the groups might also have been caused by other differences between the groups, such as differing genotypes that existed before we even did our treatment. In our example, maybe the drug doesn't actually have an effect on the mice, and group 1 is different from group 2 just because it happens to be made of mice with different genotypes.
Because people are also diverse, this is the case with humans. Because we're so different from each other, interpreting controlled experiments is problematic. So why are individuals different from each other? There are two main reasons, whether we're discussing humans or another species like these mussels. The first reason is that different individuals could have different genotypes, which could give them different phenotypes. And the second reason is that different individuals could be exposed to different environmental factors, which could also give them different phenotypes. Fortunately, individuals of model organisms are very similar to each other. But how did we accomplish this? How can we get, for example, these Arabidopsis plants to look so similar? They are the same age and have similar size, appearance, and, even initially, similar patterns of gene expression. There are two things we can do to make sure all the individuals we experiment with are as similar as possible to each other before we start our experiment. We can make sure that they're grown under the same environmental conditions, and we can use only individuals that are genetically similar to each other. Individual yeast and E. coli cells are genetically similar because they're clones of each other, because they are all derived from a single cell. For models like Arabidopsis, Drosophila, and mice, the organism has been inbred over time. yielding individuals that are homozygous for the same alleles for almost all genes, and therefore have a very similar phenotype as long as they're grown under the same environmental conditions. So again, having very similar individuals of a species allows us to do controlled experiments and allows us to say with confidence that any difference we observe in our experimental group compared to our control group was caused by the change that we made. Now, it's also generally frowned upon to purposefully mutate the genes of humans because carcinogens are substances that cause mutations, which in turn cause cancer. This means that you can't make human mutants. Model organisms might not like having their genes mutated either, but we sort of don't care as much. Anyway, it's in the name of science. We've developed many tools to easily mutate genes and isolate mutants for model organisms. For example, researchers studying drought resistance in plants wanted to determine the function of the Chi2 gene in Arabidopsis. So they started with wild-type plants, that is, those plants that don't have any mutation, and have two fully functioning Chi2 alleles, which produce functional Chi2 protein. They then mutated wild-type plants and produced plants that were homozygous for the loss-of-function mutant allele Chi2-2. And these plants did not produce any functional Chi2 protein. If you have a mutant like this, you can determine the function of the mutated gene using an approach called reverse genetics, which we'll discuss next. In reverse genetics, the first step is to isolate a mutant where the function of your gene of interest has been destroyed. In this case, we've done that for Chi2. We have a Chi2-2 mutant where the Chi2 gene is not making any functional protein. In organisms with two copies of each gene, this is done by mutating one of the two alleles of that gene such that the plant is now heterozygous. In other words, it contains one functioning wild-type allele, Chi2, and one non-functioning mutant allele, Chi2-2. This heterozygote is selfed, or if we're dealing with a sexually reproducing species, two heterozygotes are crossed to each other. Either way, homozygous Chi2-2, Chi2-2 offspring can then be isolated. And these plants are our Chi2-2 mutants. Once we've isolated our mutant, we can grow it side by side with the wild type and compare their phenotypes. In this example, Chi2 mutants and wild-type plants were grown side by side, and it was observed that the Chi2-2 mutants were less tolerant of drought. We can now assign a function to the Chi2 gene. The Chi2-2 mutant is without a functional Chi2 gene. If plants lacking a functional Chi2 gene don't grow well during drought, and plants with a functioning Chi2 gene do, then the function of the Chi2 gene is in drought tolerance. Because wild-type plants require the Chi2 gene to tolerate drought. So we could also say that the Chi2 gene is essential for drought tolerance. Because when plants don't have it, as in the Chi2-2 mutant,
They don't grow as well under drought conditions. And reverse genetics can be done in any model organism for any process being studied. For example, if mutant mice are blind, then the mutated gene functions in sight. If mutant Drosophila have white eyes, then the mutated gene functions to make eyes red, which is their normal color.
Here's another nice example of a reverse genetics approach to determine gene function, and it's from Dahl and Fergamon, 2019. These researchers isolated mutant C. elegans that had a mutated ASM3 gene and observed how long they lived compared to the wild type. Now, data analysis is an essential part of understanding genetics. So take a look at the data, take a look at this graph, take a second, and write down in 5 words or less what you think this graph shows. I'll give you a second to pause the video. Okay, so we see that C. elegans worms with a mutated ASM3 gene live longer than the wild type. Now pause the video again and answer the question, what does this result tell us about the function of the ASM3 gene? I'll give you a moment. Okay, so if worms without a functioning ASM3 gene live longer, then this gene actually reduces the lifespan of C. elegans. This seems strange, yet nature often confounds our expectations. Could research like this lead to increased human lifespans? Maybe. Who knows what the future will bring?